How to draw amino acids, the building blocks of proteins. Before we actually start our drawing in this video, like in the last video on carbohydrates and drawing the alpha glucose, let us explore the basic background on the category of molecule we are looking at, in this case, proteins. So there are four core organic molecules found in living things: carbohydrates, proteins, lipids, and nucleic acids. So we are looking at the building block amino acids of one of those four, which is proteins. So proteins, they are macromolecules or large molecules that consist of one or more polypeptide chains. What is a polypeptide chain? A polypeptide chain is just a chain of amino acids covalently bonded together. Usually it's at least 100 amino acids long. And it could be one polypeptide chain folded together, and some proteins have more than one polypeptide chain folded together to actually make the functional protein. For example, hemoglobin, it takes 4 polypeptide chains to make the hemoglobin, and it's actually 2 repeats. There's 2 alpha chains and 2 beta chains. And actually, proteins make up a significant amount of the dry weight of organisms. So if you were to dry them out and remove the water, you would find that 50% of the molecules left behind would be protein. Now, why do we care about the conformation of proteins? Well, proteins only function or carry out their jobs when they are folded correctly. When they are misfolded, they do not function as well as possible, or they might not function at all, and that can have significant consequences on the organism's health. So let's look at some of the core functions that proteins carry out. There is structural support, transport, storage, signaling, movement, defense, and catalysts. Now let's look at specific examples of each of these. So structural support, for example, your hair and your fingernails is primarily made of the protein keratin. Transport, I mentioned a moment ago, hemoglobin. Hemoglobin is a protein found by the millions inside your red blood cells, and hemoglobin protein's job is to transport oxygen throughout your body. Then we have storage proteins, storing of energy primarily. For example, egg white protein, otherwise known as albumin, stores significant amounts of energy for chick development for baby chickens. Then we have signaling. There are many different types of signaling proteins, but a signaling protein you might have heard of before would be insulin. Insulin is a protein molecule that is produced by the pancreas and released into the bloodstream to regulate your blood sugar. When your blood sugar is too high, insulin, when it binds onto your liver cells or your skeletal muscle cells, triggers those liver cells and those skeletal muscle cells to uptake the excess glucose until the blood sugar level drops down to the base level that your body tries to maintain, provided, of course, that your body is functioning appropriately. And then we have movement proteins. So we have 2 proteins, actin and myosin, that act in concert together to allow your skeletal muscle cells to contract and relax, and thus allow your skeletal muscle groups to contract and relax, and thus allow your body to move. Then we have defensive proteins. Defensive proteins include antibodies. There are different types of antibodies for every type of pathogen or disease-causing agent that exists. And when you study the immune system, you can study the complexity of antibodies. Then we have catalysts. You might have heard of the term enzymes. So enzymes are proteins that catalyze chemical reactions. They help facilitate chemical reactions. An example of that would be lactase enzyme. Lactase enzyme is an enzyme that is produced that helps cells break down lactose sugar. So people who are lactose intolerant usually produce non-functioning or do not produce lactase enzyme. And those of us who can enjoy or at least not have negative effects when we eat dairy products like milk and ice cream, that is because we have the ability to make lactase enzyme and thus digest the lactose sugar found in those dairy products. So again, these are some examples of core functions carried out by proteins. And these proteins are made of 20 different amino acids. That does not mean proteins are 20 amino acids long. It means that there are 20 different amino acids to choose from in building any given protein. So just like the English language has 26 letters, and when you rearrange the letters and you have different lengths of letter combinations, you can spell thousands of different words. With proteins, which on the short side are 100 amino acids long, when you have 20 different monomer units, amino acids to choose from, and at least hundreds of lengths of combinations. You can make millions of different proteins. So now let's explore the actual structure of the amino acid. So we're going to start off with the general or generic structure of an amino acid. So we have one central carbon, and then we have a carboxyl group, which is a carbon double bonded to an oxygen, and that same carbon bonded to an oxygen and a hydrogen. It's inferred that there is a covalent bond holding that hydrogen onto that oxygen. And then we have our central carbon bound onto an amino group, which is a nitrogen with two hydrogens. Then we have a hydrogen off of that carbon. And then what we have is what we generically refer to as the R group. Not like there's R on the periodic table, but this is like X in algebra. We change this part of the amino acid and you change the characteristic of the amino acid. You could change it and make this a non-polar amino acid. You could change it and make it a polar amino acid. You could change it and make it an acidic amino acid. You could change it and make it a basic amino acid. So we will start off with a non-polar example and move on to a polar example, and then we'll do an acidic example. If you really want to do a basic example, you will see a few in an illustration at the very end of this video. So let's do our non-polar example. So this will be glycine. It is also the simplest amino acid. So we have our central carbon, our carboxyl group, our amino group, the hydrogen, and then for our R group, we have simply a hydrogen. So there's our R group. And that is glycine. Let's take a look at a few other non-polar amino acids. So glycine, the one we just looked at, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. Now let's look at a polar example. And for our polar example, we're going to look at cysteine. So again, we have to start with the basics. Central carbon, a carboxyl group, an amino group, the hydrogen, and for our R group, we have a carbon that is bound onto two hydrogens, and then we have a thiol group, which is a sulfur bound onto a hydrogen. It's inferred again that we have a covalent bond holding that hydrogen to that sulfur, and that's a thiol group. It's one of the less famous functional groups. And so let's highlight again what makes up the overall R group. And it is the unequal sharing of electrons, specifically in this area, that gives this part of the amino acid a slight positive and negative region, and therefore makes it polar, and that is cysteine. A few more examples. We have serine, threonine, cysteine. That's the example we just looked at. Now you can see the simplification in the writing. We have tyrosine, asparagine, and glutamine. And you can see right there, that's what makes it polar. Right there, that's what makes it polar. Right there, that's what makes it polar. Polar. I think that's the part that makes it polar. I'd have to, yeah. But it might be more, this whole thing. That's not my area of expertise. Let's just erase that part. If you want to figure out what makes asparagine and glutamine polar, you can look that further up. Do not die on me, computer.